Okay, let's begin with our first lecture, that is introduction. Pathophysiology. So, what is the term pathophysiology means? Patho is derived from a Greek word which means suffering, and logos means a discourse, a system of a formal study. Any logos, like there are many different types of logos which all involve study lines, like different lines of study. You're studying about the ecosystem, then ecologos. If you're studying about the biological system, then biologos. There are all different types of logos. Okay, physio means function, pertains to the functions of the organism, so the normal functioning of a human body. Pathophysiology is the systematic study of the functional changes in cells, tissues, and organs altered by disease and or injury. So when we are studying pathophysiology, we will be studying normal aspect and what is the abnormal aspect of that system or of that cell, tissue or organ. Now what is a disease or disorder or a syndrome? A disease is a change in the structure and function, a change from the normal. There will be signs and symptoms which are evident. You can notice there will be an alteration in the homeostasis. In the normal mechanism, it is changed. What is a disorder? It's a derangement, any abnormality. It refers to a particular problem. Like when we say it is the person has a central nervous system disorder. So we know it's like related to the brain, the spinal cord. Okay, it is often used interchangeably with disease. Yes, we sometimes say we can. It is used like very casually. Sometimes it is said, you can say that the person is suffering having this disorder, or you can say that person is having the disease if you know about the disease, like that. Okay. Some terminologies which we'll be using during our studies will be acute, which means of short duration, chronic, which means of long duration, etiology means studying either the cause or the origin of the disease, idiopathic, disease that is unknown or uncertain. There will be some diseases whose cause you will not know, so those are all idiopathic. Iatrogenic, when the disease is caused by an unknown medication. Okay, it is like the person is taking almost 3-4 drugs and you cannot find out which drug did it, then you say it's an iatrogenic disorder. Nosocomial means a disease contracted by patient under medical care. Generally, it refers to all those infections which are acquired when the patient is in the medical care, when the patient is admitted in the hospital. There are two main types of cells. They are eukaryotic and prokaryotic. Eukaryotic are the ones which are found in all the humans, in those complex organisms. They have a nucleus, they have a proper cell wall, the nuclear membrane is bound, the cell wall is properly bound, all the organelles are properly formed, there is DNA in the nucleus of the eukaryote cells. What is a prokaryote? A prokaryote is like bacteria. They are all examples of prokaryotic cells. They are not so complex. They don't have like nucleus and stuff. They have free DNA in the cytoplasm. Everything is freely floating in the cytoplasm. For them, division is like, here we go, 1 and 2 divided. Very simple. For eukaryotic cells, the mitosis is like, the division is undergoing either mitosis or either meiosis, and it takes some steps. It takes some duplication of the DNA material. It takes a lot of steps, right? But for prokaryotic cells, it's a simple division, and they don't have nucleus, so no government or anything ruling over or anything. They can just divide wherever they want to. All the bacteria are an example of prokaryotic cells. This is the basic cell structure, the diagram of a cell. This central area over here is our nucleus, which will again have a nucleolus. The nucleus is surrounded by the nuclear membrane. It is the most important part of the cell because it contains all the genetic information in the form of chromosomes. This here outside the nucleus are the endoplasmic reticulums. The rough ones are the ones which contain the proteins on their surface. These granules, they are the protein granules. The smooth ones are which help in making it. Then this is the plasma membrane, which is the outline of the cell. There are some organelles which are floating freely into the cytoplasm of the cell. One of them is mitochondrion, which is called as the powerhouse of the cell. the lysosome, which which contains all the hydrolytic enzymes. The peroxisome is also one such cell which helps contains enzymes and kills the bacteria. Here they have shown cell junctions. These are the cell junctions. So what are cell junctions? They help in attaching the cells with each other. Also they help in transfer of important materials. So here is one cell junction. You can see here is another cell junction, desmosome. This one is going to attach itself to the other cell. This one is another desmosome. This another organelle is this microfilaments or cilia. They help in transporting the substances. So the cilia in the different parts of the body. The cilia in our nose helps in expulsion of the particles which we do not require. The cilia inside our body, like in our reproductive system, helps in the transport of the eggs or the sperms accordingly. This is the function of the cilia. There will also be tiny microvilli on it, which again help in transportation. This is the Golgi apparatus we know about, and this is the vesicle which is releasing all the waste, the toxic metabolites outside. Okay. Now, what are the cells differentiated? Okay, this is the when we know that a cell can perform a specific function, that means the cell has undergone the process of differentiation. Okay, that's I think one of your short question. So if you are saying that a cell can do the function of movement, then that means the cell has differentiated itself and got or acquired the function of movement. Which are the cells which can do movement are the blood circulatory cells and the maturation cells. Then there are some cells who can conduct. Which are the cells which can conduct? They are the reflexes by the nerve cells. Like when you touch a hot surface, and how do you know that you have to move your hand away if you are touching a hot surface? Then that is by conductivity with the brain. Metabolic absorption. Some cells can do the absorption. The examples are kidney cells and intestinal cells. So kidney cells help in keeping the water and the salt and maintaining the balance. Intestinal cells help in the absorption of vitamin B12 and all the other minerals and vitamins which are needed by our body. So those cells have acquired the function of metabolic absorption. Secretion. There are some cells of the hormonal glands. They can secrete. Then excretion. Some cells can do the metabolic waste breakdown and then remove it from the body. Those are the cells who have acquired the function of excretion. Respiration. So there are cells in our body like the cells of the alveoli of the lung. They can do the oxygen carbon dioxide transfer and they can help in the production of energy molecule called as ATP. Reproduction. There are all those basal cells, all the everywhere in the all the organs in our skin and all the other places. There is one layer which is a basal layer, and that layer can undergo it can reproduce. So those are called as these are the ones which help in the tissue regeneration. Okay. Communication. There are some cells which can release the enzymes through which you can communicate. So what do you mean by communicate is when the food comes, they have to communicate that okay, we need the acid to digest or break down the food, then we need a basic atmosphere so that the nutrients can be absorbed. For that is the how that is the for that function to be carried out, you require the communication skill. So that is developed by the pancreatic cells, by the cells who have been differentiated into it. One of the examples is the pancreatic cells. That was about the cell. Now coming out in the external surface of, in the outside the cell, the atmosphere outside the cell is called as the extracellular matrix. You can see over here, this is a cell, okay, which is joining together and which is forming the epithelium. Cells always rest on a basement membrane. The epithelial cells always rest on a basement membrane, and below the basement membrane here you can see the extracellular matrix. What does the extracellular matrix contain? It contains the fibroblast cells, it contains the blood vessels, it can contain the collagens, elastins, it can also contain proteins like proteoglycans and hyaluronic acid, as you can see over here. They are all linked together. So this thing is magnified and tried to shown over here. Like this is a fibroblast, and what is all the other things in its atmosphere? It is this integrins, glycoproteins, proteoglycans, the collagen fibers which are cross linked like that. Okay, and this basement membrane Like in the blood vessel also you have a basement membrane. Here in the epithelial layer also you have a basement membrane. That is magnified and shown over here. So what is the basement membrane made up of? It is made up of collagen, laminin and proteoglycan. That's why when we were studying inflammation and infection, so they contain the enzyme collagenase. Why? So that collagenase, they can break down the basement membrane and they can come over here. When there is inflammation going out and a leukocyte has to come out, it will release collagenase, due to which it can break the basement membrane and come out into the matrix. Okay, that is what is basement membrane. Now, what is the function of the cell? It can produce energy. And how does it produce energy? It has four phases. First is the digestion, where the large molecules of food are broken down into very small subunits like proteins. Proteins are broken down into amino acids. Carbohydrates are broken down into polysaccharides, monosaccharides, simple sugars. Fats are broken down into fatty acids. That's the first phase. Second phase is when the glycolysis and oxidation takes place. Like glucose is broken down into pyruvate and 6 ATP molecules are formed. That is glycolysis. The ATP molecules formed by oxidation. Phase 3 is the citric acid, which happens in the mitochondria, and here it ends with the oxidative phosphorylation in the mitochondria. And here at the end, after breaking down this pyruvate and lactic acid and everything, in the end you release CO2 and H2 and 36 molecules of ATP. So a lot of energy is released. Some of them is stored for future use, and that is how the process of energy production is carried out by the cell. Okay, now when I'm saying cell, it is not just one cell who is doing it. Okay, some cells are doing the process of digestion, some cells are doing the process of glycolysis and oxidation, and some cells are doing the process of citric acid, but most of them are doing this glycolysis and oxidation. Most of them can at least produce 6 ATP molecules for their own functioning. Okay, now how does the cell reproduce? So this is looking at the cell cycle. We all know that cells, when they are not reproducing or not doing anything, they are in the G0 phase. But if for some reason they need to reproduce, like any injury has occurred or any process has been carried out, then they enter into the G1 phase. From the G1, the cell undergoes the check. After undergoing the check, it goes into the S phase, where the DNA gets replicated. Okay, all the DNA gets, it gets doubled. Then the cell enters the G2 phase, where again a check is performed to see if the DNA that is duplicated is proper or not, or it is working properly, or it is a defective, or if there is any problem or something. After the G2 phase, the cell enters the M phase, also known as the mitosis phase. This is the phase where the cell undergoes division, ending at the phase of cytokinesis, that is division of the cytoplasm. That is the last thing to happen, right? So that is what happens in the M phase, mitosis. Then again, if the cell needs to divide, it will again go into the G1 phase. If it does not need to divide, it will go into the G0 phase, that is a resting phase. Now, what is the level of organization in the body? The basic unit is a cell. All the cells get together and make up a tissue. The tissue gets together and makes up the organ, and the organs make up our systems. So like if I'm saying the ciliated columnar epithelium, they make up the epithelial layers of the epithelial cells, make up the layer of our respiratory passage, our trachea. Then the tissue, it will, like extracellular matrix and the epithelial cells and everything, will come together and make the tissue that is called as trachea. Trachea, which forms a part of the whole of the whole organ, that is our whole windpipe, and it will further form the system that is the respiratory system. Okay, so the tissue will be called as the respiratory tissue, the organ will be called as trachea, and the system will be called as respiratory system. What are the examples of some of the tissues? They are epithelial tissue. Like I said, this basic cell get together in a line resting on a basement membrane and they form the epithelial tissue. Then what are the types of epithelial tissue? Squamous cells, columnar cells, cuboidal cells. So when I say squamous, it is more or less shaped like a square. Okay, that's why it's called as squamous. When I'm saying columnar, it is like a column, a pillar. Okay, and when I'm saying cuboidal, it is more like a rectangular shape. Okay, in 2D version, rectangular version, a standing rectangle. So that's cuboidal. The other example of epithelial tissue is stratified epithelial. So here there was only one layer, simple epithelial. Stratified epithelial means there are more layers, one above the other above the other. That's a stratified epithelial. And pseudo-stratified is when it looks like there are many layers, but actually there are not many layers. They are all just not properly arranged. So when you look, you will feel that oh, there are many layers of cells, but that is not so. They are just not properly arranged. They are arranged in a bit haphazard way. So it makes it look like it is stratified, but it is in reality not. Okay, that is called as pseudo-stratified. So these are all the terms and everything named based on the microscopic features that you see. Okay, connective tissue is another tissue which is like formed of ground substance. Then there are fibers which are collagen, elastin, and reticular fibers. That's the forming the connective tissue. The muscle tissue, which is three types: skeletal, which helps us in running, cardiac in our heart, and smooth, which is in our organs like stomach and all. They have smooth muscles. Okay, and neural tissue, which is present in our brain. So those are the main types of tissues. This is just a diagram to show you what I was trying to explain. This is a simple squamous. Here you can see cuboidal. Here you can see columnar, like columns, like pillars, right? Cuboidal, like rectangular types. Stratified is like, see, you can see different layers, one above the other. This is stratified squamous, this is stratified cuboidal, and this is pseudo stratified, stratified columnar. Okay, you cannot really show pseudo stratified because it actually is not stratified, but it looks like there are many layers. So no picture is put over here because in actual it is like this only, just simple, but it is giving a look where you are getting it confused. Okay, this is some matrix fibers, like here you can see elastin fibers, collagenous fibers, the reticular fibers, all with spatial stains. You can see all fiber, fiber, fiber. Okay, thread like. The muscle tissues are shown. You can see skeletal muscle. They are like actin myosin getting together. Cardiac muscle is more or less like this. There are less striations. And smooth muscle is very smooth. You don't really see any striations, do you? Like this. Now, what happens when something that was all about the normal cell, okay, all the normal physiological portions. Now we are coming to the pathological portions. So what happens when the cell has something changed in it? There is a stress like situation. So if there is a stress like situation on the cell, either the cell can adapt itself, if then it undergoes cellular adaptation. If it does not adapt, it can have an injury, that is called a cell injury. The injury are two types. If the stress was mild or temporary, it undergoes a reversible injury, and it can again come back to a normal cell. But if the stress is not mild, it is severe, then the cell undergoes an irreversible injury. And if the stress still continues, the cell may undergo death. Okay. This is what happens in the cell in the conditions of stress. Now, coming to the first portion, when the cell gets a stress, it has adaptation. So what is adaptation? Adaptation is a reversible structural or functional response. It is to the both normal or physiological conditions and adverse or pathological conditions. So we know stress can be normal also sometimes. Okay, not every time it is something disease related. Sometimes it is normal also. So then the ability of a cell to adapt to those conditions is called cellular adaptation. For example, like in pregnancy, it's not a pathology, right? It's a normal procedure, it's a normal process or a normal thing in life happening, but the cells are undergoing stress. Why? Because a fetus is developing inside the uterus, so the cells of the uterus have to adjust, have to adapt to let the fetus grow inside. So that is the adaptation. That is the easiest common physiological example of adaptation. What are the types of adaptation? Here you it is very clearly shown this is a normal cell. Now when the cells increase in size, they are called as hypertrophy, which happens during the pregnancy, during the in the uterus cells in the pregnancy. Some, if they increase in number, it is called as hyperplasia. Again, the uterus during pregnancy has both the features, hyperplasia also and hypertrophy also. Okay. uh, Atrophy, sometimes cells are not able to adapt, so what will happen, they will get atrophied. What happens after atrophied, they after they undergo atrophy, they can slowly start undergoing the process of death and start getting removed from the scene. Okay, so this is atrophy. Metaplasia is when there is a chronic insult to the cell, when there is a chronic, it is chronically getting insulted. So when it is getting insulted due to some acids or due to something which is not normal for the cell, it will change from one form into the other form. The commonest example is we eat food, right? When we are very starving, a lot of acid gets produced into the stomach. Now if you don't eat food at that time, the acid is going to build up in the stomach and it starts coming above, it starts coming in the esophagus. The cells of stomach are okay with all the acidic pH, all the 2 and 3 pH of the acids in our body, but the cells of esophagus are not accustomed. So when the acid starts coming into the esophagus, undergoes a stress like condition. Now sometimes in those, if it the stress keeps on happening, if you keep on having acid coming up in your esophagus, then these cells, the cells of the esophagus, which are the columnar stratified squamous cells, can undergo metaplasia and get converted into the columnar cells. Okay, that is a very common example of metaplasia. The condition is also called as Barrett's esophagus. And yes, people used to be scared because it sometime it was considered that, oh, this is precancerous. There is a controversy still going on. Some people feel that it is, and then you should start being more careful. Some people think it's okay if you remove the tissue which is metaplastic, the normal tissue can come back and it can again function properly. Okay. Now dysplasia. What is dysplasia? Dysplasia, you can see over here, this is the normal arranged cell, and look over here, these cells are not properly arranged, they are not proper, they are going in any way they want. The nucleus and the cytoplasm is also haphazard, so those are dysplastic cells, and when a cell undergoes dysplasia, it is definitely going to form cancer. Okay, so dysplasia is not good. Neoplasia, that's the second stage from dysplasia, next stage. So this is what happens, these cells underwent haywire, they went crazy, and this is the cells coming out from the basement membrane and giving the condition of neoplasia, malignancy, right? So what are the terminologies which we are going to see in this lecture are adaptation, which we just saw. Active cell injury means a cell getting injured actively. A reversible cell injury, which can reversify. If you remove the stress condition, the cell will again go back to being normal. Irreversible cell injury, when due to the constant stress, the cell has undergone an injury which cannot be repaired. That's an irreversible cell injury. Necrosis is when the cell dies. Apoptosis is when there is a programmed cell that the cell dies, but in a very well programmed manner. Chronic cell injury is when there is a constant stress like condition, then there is accumulation or infiltrations. Due to any cell injury, there are abnormal particles coming into the cell, they get accumulated. And pathologic calcification is generally when those abnormal particles that are entered into the cell, when we put them together, the calcium comes and tries to bury it, so that process is leading to calcification. Now what are the mechanisms of cell injury? Injury can be caused either by a lack of oxygen, that is called as hypoxic injury, or by a lack of blood supply, that is called as ischemic injury. You know the commonest example of ischemia, right? The myocardial infarction, heart attack, that's an ischemic injury to the muscles of our heart. Hypoxic injury happens when there is less oxygen. Okay, injury can be caused when there is reperfusion of blood also. Sometimes the oxygen, the like this is most commonly seen in our heart muscle only. Because there was no blood supply, it underwent the injury of ischemia. But then again the blood supply was given to the areas, the blood again became reperfused. Then because of again sudden flow of blood into the tissues, it can lead to a injury condition that is called as injury due to reperfusion. It is why is it caused is because of the sudden flow of the blood. Suddenly the muscle cells don't know what to do, and because of that, free radicals and reactive oxygen species and those molecules start getting formed into the body, because the process of metabolism is not going on properly, because the cells were undergoing ischemia, right? So this is all leading to even more cell injury. From that, I'm jumping to the topic of reactive oxygen species. It is one of those concepts which you should know. Like I included Zika virus in infection. Why? It's not like it's important, but you should know. Some portion of his knowledge is important. So reactive oxygen species, they are made up of atoms of oxygen and hydrogen. It is formed due to the injury by radiation or chemicals or UV light, due to the chemical reactions going wrong, and so positive and negative charges remain on them. Like the oxygen and hydrogen molecules, they generally get neutralized, right? But if there is like a chemical injury or a UV light injury, then the positive and negative charges remain on those molecules and they get converted into a reactive oxygen species. What are the examples? Superoxide, hydrogen peroxide, hydroxy radical, they are all examples of those free radicals. They can affect the lipids, proteins, and even the DNA material. That's why they are something which we are very scared of, because they can affect the lipids. Because they affect the lipids, the cell membrane of all the cells start getting affected. Because they can affect the proteins can undergo misfolding, and then they can lead to formation of defective enzymes, and the different defective functions start getting formed. If they affect the DNA, it can lead to mutations. The mutations, when that keeps on happening, the cumulative effect can happen, and the person can develop a malignancy. Or because of the mutation, they can also have some enzymatic deficiency. Okay, yeah, this is the diagram shown. Superoxide, hydrogen peroxide, hydroxy radical, these are the reactive oxygen species. What are the effects? Lipid peroxidation, membrane damage. You see, protein modification, breakdown of the proteins, the DNA damage, the mutations that are happening. Okay, normally what should have happened? Normally the O2, the superoxide, should have converted into H2O2 by the superoxide dismutase enzyme, and it should have been removed. The H2O2 should have been converted, decomposed into H2O, and should have been removed from the body. Okay, but it is not getting converted, and so this gets accumulated. That is the reactive oxygen species. Okay, what is the general mechanism? Now we are going back to the cell injury. Remember the ischemic injury, hypoxic injury. Now what are the other ways that the cell can have injury? It can be either by some chemicals or toxins. It can either be the gases like carbon dioxide, sulfur dioxide, nitrogen dioxide. All those pollutants in our air can also cause cell injury. Then there are heavy metals like lead, mercury, ethanol, cadmium. They can also cause cell injury. So all those people working in the industries which are dealing with lead and stuff, they are supposed to take extra precautions. Why? Because the exposure of lead can cause cell injury, and due to the constant cell injury, the cells can undergo dysplasia, and later on they can also develop the neoplastic features, right? So that is cell injury. Social and street drugs like cocaine, marijuana can also injure the cell. Then even physical injuries like asphyxial, strangulation, drowning, that can also cause cell injury. Other agents which can cause cell injury are the climate change like extreme heat, extreme cold temperatures. They can also, because of changes, they can affect our skin cells, and they can affect the cells like we are breathing those cold and hot airs. So they can affect the cells of our body also. The UV light, the ionizing radiation, they can also affect the cells of the body. I had already discussed this ethanol metabolism. I'm just trying to remind you again what is exactly happening. The ethanol gets converted into acetaldehyde with the help of ADH enzyme. Aldehyde, aldehyde, acetaldehyde dehydrogenase enzyme. Okay, then acetaldehyde gets converted into acetate by ALDH2 enzyme, which is okay. So this is acetate dehydrogenase, this is aldehyde dehydrogenase enzyme, and it gets converted into acetate. Acetate are converted into ketone bodies, fatty acids, and amino acids, which are later metabolized into CO2 and H2 and expelled from the body. Now, if due to some reason acetaldehyde is not converted into acetate, then it can cause protein adducts, or it can also cause problems of lipid peroxidation. It can also cause stress like condition. So that way it can cause cell injury. So that is how drinking causes injury to us, to our cells, right? What are the manifestations of the cell injury? There are two manifestations. Either there will be accumulations as the result of cell injury, or the cell can die. Cell can die by the process of either necrosis, apoptosis, or autophagy. Autophagy generally means it kills itself. Intracellular accumulation. Now, when I say intracellular accumulation, these are some of the examples. What happens when there are intracellular accumulation? Here you see fat cells getting accumulated in the liver because of constantly we are eating all those fat food, junky foods and stuff like that. If it does not get metabolized properly, it starts accumulating in the cells of the liver like this, like the droplets, and leads to a condition called as fatty liver. And what you see when you see it under the microscope, you see fat droplets in the liver cells. Okay, that's one example. Another example is sometimes proteins getting mutated and they get accumulated in the cell. So there you can see there is an accumulation of a lot of abnormal proteins. Okay, sometimes the enzyme which is supposed to help with the digestion is not present, and because of that you can see diseases like lysosomal storage diseases, because of which there will be accumulation of a lot of material in the cell. Okay, the last example is of a ingestion of the indigestible material. So what it means is sometimes some materials get into the cell. Like easiest example is I can give of when you are breathing polluted air, there is coal particles in the air. They are so tiny that even the cilia cannot expel it out. So those coal particles start going inside and they go into the lungs. When they go into the lungs, the cells of the lungs absorb them and they stay over there. So that's why when you see like it is the pollution level over here is very less, but if you see in India the lungs of the people, they have black spots in them because of the pollution, because of all those coals pollution and all the smoke. There are black spots on the lungs because those coal particles get deposited in the cells of the lungs. Okay, so that is accumulation of indigestible materials. Same thing is also seen in some occupational hazards. Okay, those people who are working in glass industries, the glass particles are so tiny that they cannot be removed and they get deposited into the body. Okay, leading to some conditions like silicosis. If they are working in asbestos factory, asbestosis, stuff like that. Those are one type of intracellular accumulation. What are the other types of intracellular accumulation? When lipids and carbohydrates get accumulated, the disease is called as lysosomal storage disease. Fatty changes in the liver, Niemann-Pick disease. When glycogen gets deposited, it is called as glycogen storage disease. When protein gets deposited, the condition is called as Russell bodies in the multiple myeloma, or in Alzheimer's disease you see those neurofibrillary tangles. So even in Alzheimer's disease, if you see the brain cells, you will see that there are protein depositions. This is the other kind of degeneration known as hydropic degeneration. So what happens sometimes is when there is hypoxia, less oxygen, the ATP production decreases. Because of that, the sodium and water move into the cell, and potassium, which is supposed to stay inside the cell, that comes out. That because of that, the osmotic pressure increases. As the osmotic pressure increases, even more water starts coming into the cell. Now that water is abnormal. The cell does not require that much water. So that water starts coming into the cells and starts forming vacuoles like this, and as they start forming vacuoles, they start affecting the other organelles of the cell like endoplasmic reticulum, and they swell up the endoplasmic reticulum, and even it starts undergoing bursting, and there is a lot of vacuoles or water vacuoles filled in the cell. That extensive vacuolation is called as hydropic degeneration of the cell. It generally is seen in conditions of hypoxia. What is calcium deposition? There are two types of calcium deposition. One is dystrophic, one is metastatic. Dystrophic is when the cells have died at a place and the calcium gets attracted to the place locally, and it forms like calcium deposits, tiny calcium deposits at the places where the cells have died. That is called as dystrophic calcification. Metastatic calcification is seen like in some malignancies, and not in some, in most of the malignancies the calcium levels become raised because of certain mechanisms which are very complicated to be understood or explained in this lecture. So I can just say this, that in the conditions like malignancy, the calcium levels are raised. Because of the raised calcium levels, they can lead to deposition at any place in the body, leading to a condition called as metastatic calcification. Okay. Now, what are the systemic? How will our body manifest that the cells are undergoing injury? So the person will have fever, there will be increased heart rate, there can be increase in the number of leukocytes, there can be pain at the site. These are all the manifestations how you can know that, oh, the cell injury has happened. What are the enzymes which also indicate us or help us in leading that, okay, the cells are dying? So there are these enzymes like CK, creatinine kinase. If there is any injury to the skeletal muscle or the brain or the heart, then the CK enzyme is elevated. LDH is the most non-specific enzyme, and it is always raised if there is somewhere some injury going on. Okay. EST is limited to your heart, liver and skeletal muscle. ALP is related to the liver and bone. ALT is related to the liver, kidney and heart. Amylase is related to the pancreas. Troponin is related to the heart. So that's why this is helping you. This will give you an idea that whenever you are suspecting myocardial ischemia or myocardial infarction, and not only is ECG done, some enzymes are ordered. So what enzymes are there? They are troponin and they are CK, because they, and ALT. Why? Because they, if their levels are raised, it will give you an idea that yes, heart muscles are dying. Yes, ischemia has taken place. Okay. Now this is necrosis versus apoptosis. So what happens in necrosis? We have already discussed this. It is the dirtiest form of cell death. Okay, you see over here the cell has undergone swelling, there are membrane blebs getting formed, more and more injuries happening, and because of this everything starts disintegrating. There are inflammatory cells which are coming and breaking down all the parts of the cells, and the mitochondria is also undergoing breakage. So this is all leading to a condition called as necrosis, a dirty way of the cell dying. Okay. The apoptosis is, everything is very programmed, very specific. So there will be condensation of the chromatin, the membranes will form tiny blebs, those blebs will start fragmenting and they will start dying themselves inside, they will start killing, so that a lot of reaction doesn't happen around, and it will be a very clean death. And after this apoptotic, these cells start getting separated, they will go into the macrophage or any phagocytic cell and die inside that cell. So very least amount of, very least amount of wastage is coming out over here. Here you can see everything wastage is coming out over here. So because of that, necrosis is more dirty, the body shows a lot of symptoms, and apoptosis body generally does not show a lot of symptoms. Okay. Now coming to the next portion of the lecture, it is about the fluid. So there is a balance maintained by our renal, kidney, by the hormones and by the nervous system. So because of the balance maintained, 2/3 of the fluid stays intracellular and 1/3 stays outside, extracellular. Now when you're talking about the 1/3 extracellular fluid, which fluids are included in those? there is the interstitial fluid, the fluid between the cells. Then there is intravascular of the blood volume. Remember the blood plasma, 5 liters. So that's intravascular. That, but it is a part of extracellular. Okay, it's not inside the cell. It's in the blood system, right in our vessels. Then other lymph, like all the lymphatic systems. Then transcellular fluid, like in the pleural in our lungs, around the lungs, in synovial in around our joints, peritoneal is in and around our stomach, abdomen, pericardial is around our heart, and intraocular is in our eyes. These are all the other places where you can find the fluid, the extracellular fluid. Okay, the fluid or the flow of fluid is maintained by Starling's forces, by two main pressures, hydrostatic and osmotic, or also called as oncotic pressures. Okay, so we have to know what are the pressures and what direction are they acting. So the tissue hydrostatic pressure is trying to push the water inside, as you can see over here, so that the sign is this way. But the tissue oncotic pressure is trying to pull the water. Okay, the here you can see the capillary stage. This I'm only discussing at the capillary stage. Okay, why? Because at the capillary stage the diffusion is taking place. In the arteries the forces will change. In the arterioles, arteries, in the big vessels the forces change direction, but they, in the basis they remain the same. Okay, now here you see capillary hydrostatic pressure is trying to exert a pressure towards outside to let the fluid come out of the capillary, and the capillary oncotic pressure, because of its particles inside the capillary, it is trying to pull the fluid inside. Okay. This four are always in balance so that everything goes out properly. Imbalance in any of these pressures can lead to edematous condition or any other pathological condition. So one of the commonest example is edema. How does edema happen? So what happens is, either there is a decreased synthesis of the plasma proteins, or there is a loss of the plasma proteins, or there is a lot of water retention in the plasma. So what will happen because of that? There will be a decrease in the capillary oncotic pressure. Because the capillary oncotic pressure, you see over here, the capillary oncotic pressure is decreasing, so the fluid which is supposed to come inside the capillary does not come inside, it stays over here in the tissues, leading to a condition known as edema. Okay, the other thing which can happen is there is an increased capillary permeability, like in burns or in inflammatory condition the capillary permeability increases. Because of that the plasma proteins start getting out of the capillaries. Because of the loss of the proteins, again there is a loss of proteins, so the capillary oncotic pressure decreases, leading to a condition known as edema. Okay, now not only does the loss of plasma protein cause a decrease in the capillary oncotic pressure, it also increases the tissue oncotic pressure. So if as it increases the tissue oncotic pressure, you can see over here it is trying to take the fluid outside. Okay, so because it is trying to take the fluid outside, it is in fact increasing or helping in increasing the edematous condition. Okay, what else happens? There is a decreased transport of the capillary filtered protein. There is a lymph obstruction which can lead to this, and the lymph obstruction can also lead to a fluid movement into the tissues, leading to edema condition. There is increased capillary hydrostatic pressure because of any venous obstruction or salt and water retention or any heart failure like condition. So again I am trying to show you the increased capillary hydrostatic pressure. It is increasing, so the water, they are pushing the water outside. Again it will lead to fluid in the tissue, leading to edematous condition. Okay, basically all these four forces should stay in balance. If any of them increases or decreases, it is going to lead to a condition called as edema. Okay, now coming to the last segment of the lecture, it is about the electrolytes. I have not included a different slide like what is the cause of hypernatremia, hyponatremia, cause of hyperkalemia, hypokalemia. I have not done anything like that, but I have given an overview of the electrolytes. So we'll do the overview.